Hello everyone, I'm Saida. I welcome you all to my presentation on unintentional release of antibiotics associated with nutrients recovery from source-separated human urine by biochar. When we hear the word human urine, we think of a waste, which is actually mixed up with the municipal wastewater system and sent to the wastewater treatment plant. But this urine also contains some significant compounds like ammonia and phosphate. In a study have shown that human urine contains ammonia at a concentration of 1.79 to 2.61 gram per liter and phosphate at 0.21 gram per liter. These two compounds is actually plant essential nutrients. So as a matter of fact, we can say that urine can be used as a plant nutrient supply in agriculture. And if we can separate this urine at source, we can have more concentrated form of nutrients. So we can say that the dilution of these nutrients with the municipal wastewater system is not a good solution. However, this is not the only compound present in urine. Actually, every day we intake some kind of medicines, and these medicines after metabolism into our body goes directly to the urine and make their presence. Previous studies have shown that the presence of ciprofloxacin, sulfamethoxazole diclofenac, and trimethoprim at microgram per liter level in the urine. So to avoid this, people have been applied biochar as an adsorbent to recover nutrients from human urine. They have hypothesized that human biochar will uptake ammonia and phosphate from the urine, and then this biochar can be used as a slow releasing fertilizer. However, researchers have also again overlooked the fact that biochar is an adsorbent. It can also adsorb pharmaceuticals and other contaminants present in the urine. In this study, we have only considered the antibiotics as a contaminant in urine. So these antibiotics need to be considered prior to the application of source-separated urine as a nutrient product. So the main question comes here, will the presence of antibiotics be a problem? We are assuming that if we apply biochar as an adsorbent, it will uptake antibiotics, but is it true or not? So the objective of our study is to examine adsorption of antibiotics and nutrients in source-separated human urine by biochar and also subsequent desorption, because if the biochar is uptaking antibiotics and when we are applying this biochar as a fertilizer, there is no desorption of antibiotics, then that is not a problem. So the desorption is also an important fact here. The hypothesis of this study is that the biochar can absorb antibiotics while recovering nutrients at a wide range of pH by electrostatic interactions. And the second hypothesis is antibiotics absorbed on biochar can also dissolve. First, we have collected source-separated urine, and then we have applied biochar into the urine. A specific pharmaceutical has been spiked into the urine solution, and then biochar loaded with nutrients and antibiotics is been filtered out. After that, this biochar has been taken is been dissolved into aqueous solution to see that there is any desorption of antibiotics. The more the desorption, the more will be the environmental degradation. For this study, we have chosen two different types of biochar: oak wood biochar pyrolyzed at 900 degrees Celsius and peppermill sludge biochar pyrolyzed at 400 degrees Celsius. The kinetics and isotherm studies have also been performed at biochar dose of 0.1 to 20 gram per liter, pH ranging from 1 to 11, and adsorption time of 0 to 24 hours. In this table, we can see the properties of biochar, and the properties of biochar actually vary depending on different factors like pyrolysis temperature, the type of biomass we use for producing biochar, heating rate. Here you can see that the OW biochar have a much higher carbon content than PMS biochar. However, the PMS biochar have more hydrogen, oxygen and nitrogen content than OW biochar, and also the surface area of OW biochar is much higher. This two figure here is showing the FDIR adsorbance of the biochar before and after adsorption. So here the phenolic, OH, the CC, the CH, the CO bond are showing the presence, showing the results of the proof of adsorption of antibiotics and nutrients onto the biochar at different wave number. The first result here is showing the effect of pH on adsorption. In the y-axis, you can see the percent removal of compounds, and the x-axis we can see the pH from 1 to 11. Here, both of the biochar have shown the similar trend for antibiotics. The pH, the removal is decreasing with the increase of pH, whereas the removal of nutrients is increasing with pH and then decreasing. The same thing we can observe here, but with a different amount. Like the antibiotics is decreasing with pH and the nutrients is increasing with pH. So in summary, we can say that acidic pH or pH less than 5 is good for antibiotics removal, and basic pH greater than 5 is good for nutrients recovery. Here we can see the adsorption kinetics of nutrients and antibiotics on biochar. From both biochar, we can observe almost a similar trend, and the adsorption equilibrium reached almost at the same time. However, the amount of adsorption of nutrients is much higher than the amount of adsorption of antibiotics. This is true for both biochars. The PMS biochar is absorbing more ammonia and phosphate than OW biochar. However, OW biochar is uptaking more antibiotics. This study have been also done on granular activated carbon to show people that this thought or this concept is not unique to biochar. This can happen to any other adsorbent applied to the urine. You can see here that the granular activated carbon is also uptaking nutrients and antibiotics at the same time. However, in comparison with biochar, the absorption of GSE is much higher for both nutrients and antibiotics, but GSE required much more time to get to reach the equilibrium than the biochar. This diagram here is showing the desorption of nutrients and antibiotics. Here you can see that the desorption of highest desorption was observed for TMP, and the lowest desorption was observed for sulfamethoxazole. So do we want desorption or not? For nutrients, yes, we would like to have some desorption. But for antibiotics, no, we don't want to have desorption, because the more desorption of antibiotics means more contaminant are released into the environment. So in conclusion, from all the results we have shown here, we can say that simultaneous adsorption of nutrients and antibiotics were observed at a wide pH range. The results of our work could serve as a building block for future research to find a way for separating nutrients and antibiotics by adsorption. The future recommendation from this study is to use some surface modification to the biochar to improve nutrient and antibiotic selectivity to recover and reuse nutrient in human urine in an environmentally sound manner. And in the beginning, we have said that antibiotics is not the only chemical entity present in the urine. There are some other contaminants too. So the future study could also consider the presence of other contaminants in urine, such as disinfectants and pathogens. Thank you.